Assalamu alaikum, everybody. I hope you all are doing well. My name is Zaid Al Sarawi. I'm a first year medical student, and today I'm going to be teaching you guys the MOL TBL crash course, inshallah. I know the amount of material we have is overwhelming, but inshallah, this crash course will help make it easy. Inshallah, you guys have a safe and smooth journey while studying this. And yallah, well, let us begin. Before we begin, actually, I want to thank my good friend Taha Sayed for helping me with the slides. He has been a great help, and yallah, let us begin. So first of all, I want to just let you guys know that we have almost like six lectures, right? But we can say they're possibly five because 22 and 23 are literally like almost one lecture. So yeah, and the only lectures you're gonna really need to like really actually focus on is 20, 21, 22, 23. They are very high yield. 18 and 19 build concepts upon these. So yeah, let's begin. So let's begin with the disposal of nitrogen. So as we know, let's get some background information on this. Unlike carbs and fats, our body actually never stores amino acids. They never store it, and actually, it's really all excreted in urine. So this is something we need to just realize and familiarize ourselves with, because our body just never really needs to store and keep our amino acids in our body. We're only using them to synthesize our tissues, and then eventually, after we synthesize like tissues, we're just going to excrete it, which is why we may be able to obtain amino acids like from a few sources only. And these sources may be dietary sources, de novo synthesis. De novo synthesis just basically means your body is making it, you know? And finally, protein breakdown. Now, if you guys notice, whenever we eat food that's rich in diet, like, protein, we're technically like, having it dietary sources, and then we're having the protein breakdown, right? Which is why this is all called protein turnover. Protein turnover is basically just like proteins that are simultaneously degraded and resynthesized. So that's just a concept I wanted to clear. And which is why it reaches to the eventual eventual expedition that we need to realize that nitrogen also, in some cases, right, it is, or in most cases, sorry, it is balanced, okay, between synthesis and excretion. But however, there are some cases when we come to realize that nitrogen may not be balanced. Sometimes there's we may have something called a positive balance. Maybe sometimes you have something called a negative balance. Now, positive balance basically is when you're after illness, okay, where you have the storage of more nitrogen than excretion of nitrogen. So the way I'd like to like, remember this is by cortisol. After we're like, sick and after we recover, our body is basically more positive. We're feeling much better, more ecstatic, and we're having more storing than excreting, But when it's a negative balance, this is like during illness, trauma, stress, our body is like, excreting more than storing, and it's rushing to push out all of this, nitrogen. That's the way I'd memorize it. I hope this helps. So, next slide will be talked about amino acid catabolism. Now, for us to understand also nitrogen, okay, we need to understand that 80% of nitrogen, it comes from the urea cycle, okay, which we will discuss later. 3% will come from the kidney in the form of ammonium. Okay, just familiarize yourself with this. And also, our body, the reason it rushes to excrete nitrogen in the form of ammonia or from urea is because ammonia itself is a highly toxic material, and it either has to be removed in the form of free ammonia, which is NH4 plus, or urea from the urea cycle. So let us begin by familiarizing ourselves with aslan the amino acid structure. This is the famous amino acid structure. We already know it, but I just wanted to like get you guys kicked up again on what it is. Okay. So first of all, we have this side, left side. This is called your amine group or alpha amine group. It is basically NH2. This is the main guy over here we're going to talk about, by the way. We have the carbon connected to hydrogen and the R side chain. And then we have COH on the right side. And our main objective is going to be NH2. Now, as I told you, this is basically just discussed. And yeah, it is attached basically to a central alpha carbon except proline. Basically, you have alpha amino groups and which is, all your amino acids except for proline, okay? So the presence of the alpha amino group, which is your NH2, it keeps the amino acids protected from oxidative breakdown. Basically, what this means, our body, Sahwi said we're going to use this amino acids, to synthesize tissues and other stuff. our body wants to preserve أصلا the amino acid, so we keep NH2 in order to synthesize and be able to utilize the amino acid rather than let it break down. So the removal of the alpha amino group is required for amino acid energy production and other stuff we're going to talk about. And this is done by two main important steps of this lecture I wanted to focus on. Transamination and oxidative deamination. We're going to get to it right in a second. I just want you guys to recognize these two because we're going to talk about them now in depth. And finally, as we said, this produces ammonia or ammonium. It doesn't matter. Both are the same form, but just recognize both. And yeah, this is basically the main guy of the lecture that we're going to be focusing on. So amino acid catabolism process. Now let's actually get into the process, which is beginning transamination. Transamination occurs basically when you have your amino acid, right? And this is called something your alpha ketoglutarate, right? Our transamination, we're gonna we're gonna move it, transmit it, transmit the amino group to the alpha ketoglutarate, and this is done by, your alpha amino transferase. And when it transfers it, you're going to be left with something or like new byproducts. So we're gonna talk about them. The alpha amino group may consist of amino acids that transfers to alpha ketoglutarate to form glutamate, okay? And this is your first byproduct. And yeah, they basically amino transferase transfer alpha amino group from an amino acid to alpha keto acid. So your alpha ketoglutarate is also considered an alpha keto acid, and it becomes a new alpha keto acid eventually. While your alpha ketoglutarate, this one, right, it becomes the glutamate, while the amino acid without the NH3 becomes a new alpha keto acid. It's basically just switching roles. You can think of it that way. You know? Anyways, apart from that, we have oxidative deamination. Oxidative deamination, by starters, we have here, after we talked about the alpha ketoglutarate, right? And then we have the amino acid, it becomes glutamate. But eventually, glutamate will undergo oxidative deamination to form NH4 plus, which is ammonium, which is what we said also in the byproduct, okay? So yeah. And also, these alpha keto acids may be pyruvate or oxaloacetate, depending on which enzyme is used. So basically the alpha keto acids that we already discussed about, right? After the transfer of alpha ketoglutarate and all of that, they may end up as other byproducts, and we're gonna talk about it in a second. And basically, summary, it yields alpha keto acid or like a new alpha keto acid and NH4 eventually through glutamate. Okay. Let's continue and let's talk more specifically about transaminases, which we just discussed the first step. So what are transaminases? Transaminases are basically enzymes, okay? And these enzymes, they transfer an amino group from an amino acid to an alpha keto acid, also called your alpha ketoglutarate, right? So yeah, and examples of transaminases, which we talked about, like the enzymes, you know, are alanine aminotransferase, okay, which you guys have probably heard about in the lectures about like, clinical enzymes or clinical enzymology, in the first semester. Sorry, the pre-midterm, and we're gonna talk about them more in details, So alanine aminotransferase, also called ALTs, basically take alanine and this alpha ketoglutarate. So here you have the amino acid, we have the alpha ketoglutarate. Your alanine is gonna remove the NH2 and give it to alpha ketoglutarate to form glutamate, right? And however, alanine is gonna eventually, without the NH2, is gonna become the pyruvate, okay? The second example is aspartate aminotransferase. Basically involves aspartate and alpha ketoglutarate. However, it will form oxaloacetate and glutamate. Both of them will form glutamate, but I really need you guys to recognize that AST is going to form oxaloacetate. Alanine aminotransferase is going to form pyruvate. Okay? So why is this all important? This is mainly important because glutamate itself acts as a temporary source of storage of nitrogen for amino acid synthesis eventually, because we need to store the nitrogen so we don't leave it, going in the body and becoming harmful. Now, something I want you guys to recognize that the role here, okay, ALTs and ASTs both are very important for the production of glutamate. This is like a common rule, okay? But ASTs actually, if you notice, both the reactions are reversible. ASTs follow a reaction more commonly reverse. Basically, it starts as a glutamate and oxaloacetate and then forms aspartate and alpha ketoglutarate. Now, we ask Zaid, why is this the case here? The case here is because aspartate itself it needs to form aspartate from oxaloacetate, and aspartate is going to be used for the nitrogen, or like, to carry the nitrogen, right, into the eventual the urea cycle. It has its own cycle. We'll talk about it, but basically just understand the aspartate aminotransferase. It has its own, like, it doesn't move like alanine, from here to pyruvate. No, it mainly prioritizes aspartate formation for the urea cycle. Okay. Now, obviously, we have some things, right, that are supporting the aminotransferases or aminotransaminases here, right? And these cofactors are like PLP, which is basically a derivative of B6, and it's the main cofactor supporting it here. And finally, as I just told you guys, elevated ASTs and ALTs are seen during liver diseases, and also they are seen in other myocardial diseases. And finally, here's like the reaction, just more in detail. You have the ALT, and PLP together to act upon, like, or AST and PLP to form upon the transaminases. And yeah, that's about it. Now, let's talk about oxidative deamination. Oxidative deamination, okay, it is like a process that's mainly seen facilitated by glutamate dehydrogenase, okay? So we talked about the transaminases like ALTs and ASTs that function as the enzyme there, or like, what's supporting the formation. Here in oxidative deamination, it's glutamate dehydrogenase processing this. And it's basically, it takes glutamate as the most important factor as transmitting amino group eventually into NH3 and alpha ketoglutarate. Now you'll be like, Zed, didn't we already, say that it becomes an alpha keto acid like pyruvate, oxaloacetate? Yes, but there, in some cases, it is actually going to regenerate alpha ketoglutarate, so it can reuse it in the cycle. I'll talk about that in a second. But let's talk about, we said there's PLP as a cofactor. Oxidative deamination also has two cofactors here. The cofactors are involved are NAD plus and NADP plus. Okay, and this is actually the only enzyme that utilizes both of these. So yeah, just keep that in mind. Okay. Also, glutamate activity, and all of amino acid degradation. All of this level, the levels are high, and they're mainly high during like two stages, like after a protein-rich meal, because obviously we're degrading protein, and also when energy levels are low. If you guys remember in the insulin lecture, we talked about how we use fat, carbs as energy, but then eventually we're going to use proteins. Our body doesn't eventually use proteins as our energy source. But if energy levels are very low, they're going to begin using protein, you know? And yeah, that's basic tells you these energy derivatives are mainly from the carbon skeleton breakdown. And you'll be like, what does this mean? If you remember in the alpha, after we remove NH2 and we have the leftover amino acid, this amino acid becomes the carbon skeleton eventually, quote unquote. And this carbon skeleton eventually is gonna, like, prioritize for glucose synthesis, energy synthesis. And yeah, that's about it. So as you can guys see here, this is the transamination process we already talked about. NH2 is, transaminating is moving to the alpha ketoglutarate. Alpha ketoglutarate comes here. It will form eventually, your alpha keto acids, and your NH is going to form, NH of the glutamate instead of the alpha amino acids. And then they're going to move, NH2 with NAD plus as a cofactor. They're going to undergo oxidative deamination to form NH3 as a byproduct, regenerate NADH, and also to regenerate alpha ketoglutarate, and then to continue the cycle. And yeah, this is basically just explaining it more over. And yeah, also a very important thing I want you guys recognizing, which is again, is the carbon skeleton concept I just talked about. So please understand, because we're going to talk about it more later. So let's talk about the glucose alanine cycle. So I hope I broke this down well for you guys, because wallahi, when I was studying this, it was kind of hectic. But still, inshallah, it's understood now. So first of all, the glucose alanine cycle, okay, takes place between two organs, or like, two structures here, the muscle and the liver, right? And the muscle and the liver, it will explain how we, receive all the processes that we just talked about. So for starters, we have protein. Okay. Protein, after we're going to take it, we want to break this down. So, right. So protein is going to eventually become amino acids. After we break down amino acids, or sorry, protein into amino acids will then undergo the steps we talked about, and it's going to form eventually ammonia, right? Oh, wait, sorry. Protein to amino acids, it's by protein degradation. And then eventually you're going to have NH4 plus, through the two steps transamination, oxidative deamination we just talked about. And then surprisingly, okay, NH4 plus, okay, I want you guys to remember this. This is not a concept that's spoken about a lot. NH4 plus is gonna actually form glutamate. Now we're like, how does this occur? Isn't NH4 the last step? Yes, but it does it through a very important step called reductive amination. Basically, take oxidative deamination and flip it. Oxidation, reductive, and deamination, amination. Now this is low yield, but it still is something to understand. So, really good to understand. So why do we convert glutamate to that type? We want to take glutamate and pyruvate. If you guys notice, this is similar, which are the byproducts, if you remember, of our ALTs, and then it's gonna form eventually alanine, right? And it's gonna form alpha ketoglutarate. Now, the reason also we need, this step to, regenerate glutamate is because you want to form alanine. Now, alanine is important. We'll talk about it in a second. And basically, it just uses, ALTs or alanine aminotransferase, and then it will continue through the blood and travel as blood alanine, eventually arriving on the liver itself, right, with alpha ketoglutarate, okay? And then it's going to actually regenerate pyruvate and glutamate, okay? And this is actually done through also, ALTs, obviously. And the reason we do this is because our alanine actually temporarily carries, right, the ammonia in like a safer form. So, this is why we convert, NH4 to glutamate, the glutamate to alanine, because we want to carry the ammonia in like in a safe place. And then eventually, when we arrive at the liver won't, it carries ammonia in the form of, or sorry, carries nitrogen in the form of glutamate. And then eventually glutamate will form NH4 plus, and then after that, it's gonna go to the urea cycle. So what happens afterwards? So we want to regenerate all of this. So this is where the regeneration is going to occur. Pyruvate will form glucose, and this is through gluconeogenesis, obviously. And then glucose will travel as blood glucose. This is familiar. And then it's going to eventually end up as glucose in the muscle, where it will undergo, glycolysis to reform pyruvate. You know? And yeah, that's about it. So here in most tissues except muscles, ammonia is transported. Sorry, not it's something here I mentioned that I need you guys to recognize. Glutamate, it carries nitrogen, not ammonia. Glutamine, okay, which we'll talk about right now, basically carries glutamate. Glutamine carries glutamate and ammonia. Okay, so basically done mainly by glutamine synthase, and it combines both glutamate and ammonia. And it's just important to emphasize because it carries it and it ensures that NH4 plus isn't harmful. And basically at the liver, it returns it back to glutamate and ammonia. So there's a temporary stage where it forms it, and then it reforms it back to glutamate and ammonia. You know? Anyways, so just as this review again, it basically has a temporary storage of nitrogen. Glutamine has a temporary storage of ammonia in most tissues like your liver. However, alanine, it has a temporary storage of ammonia in muscles, okay? And yeah, that's about it. Just remember these and you'll understand, I guess, the concept there. So let's talk about the in-house proteins, okay? Now, in-house proteins, we've already taken this in MOL 1, but let's go over it again. So some proteins, right, they can be misfolded, and this is bad for our body, obviously, right? So to cause, proteins, to prevent, from being misfolded and defective, we have something called ubiquitin. Ubiquitin, it helps prevent that by a series of steps I'm going to talk about. So for starters, ubiquitin, okay, it has a terminal glycine. It binds to lysine residues on the protein via isopeptide bonds, and basically it requires ATP here. Okay. And then after that, it binds to the defective protein we have. You're gonna have something called the 26S proteasome, and this is basically a complex that helps degrade, right? Or which is responsible for degrading eventually your defective protein. We're gonna talk about it in a second. And basically recognizes and binds to the tagged protein. This is also ATP required. And protein is degraded into 7 to 8 amino acid peptides. And we don't degrade it fully because eventually we're gonna just break it down even more, through the steps of transamination and all of that. And then also finally, ubiquitin is released and recycled. So yeah, that's about it. Here, I just want to show you guys that ubiquitin with the terminal glycine, it will form the isopeptide bond. And then you also have this is the target protein. You'll have, apart from that, the 26X proteasome. It's not really shown here. But basically it binds to the attack protein and acts as a complex to, degrade it into amino acid peptides. Yeah, and then free amino acids eventually. And yeah, this is just basically a way to remember it. That this is your, this is your misfolded protein, and here you have your ubiquitin attaching to it. So let's talk about the final thing here, which is our clinical applications for this lecture. They're very easy, by the way. I hope I made them as concise. The first clinical scenario we want to talk about is cystinuria. Now, cystinuria, okay, basically occurs when we're gonna notice excess amino acids, and these excess amino acids we're gonna notice is cysteine, ornithine, lysine, and arginine. And I just did it in the mnemonic cola, so I hope you guys remember it. So just basically know that you have excess amino acids. You're gonna see them in urine. Okay, and yeah, this is just to remember it. This is an autosomal recessive disease. And basically, inabsorption of these amino acids leads to the problem occurring in large amounts in urine, causing and leading to kidney stones, which is basically due to cysteine. Okay. So, kidney stones occurs basically mainly by cysteine. I want you guys to remember this. Okay. And basically, you're going to notice these excess amino acids. Our kidneys, obviously, and also small intestines have specific transporters for amino acids, if you guys remember in GIT. And the types that we need to know here is that type 1 heterozygotes basically look normal in urine tests, and types 2 and 3 heterozygotes leak extra cysteine and amino acids. So basically, if which one's more dangerous, obviously type 1, because you might not know that you have cystinuria by the shelf. The second clinical application we'll talk about is called Hartnupp's disease. Now, Hartnupp's disease, basically, here we talked about cola and the amino acids. Here we have actually one amino acid that's very important, helps with the kidney, right? And it's basically tryptophan. Tryptophan is an important amino acid that helps with the kidney eventually, and then basically it's found deficient in Hartnupp's disease and the kidney, and basically presents in urine in large amounts. Basically, you have neutral amino acid transport defect that occurs, just to summarize it. And because of that, no tryptophan means no vitamin B3. And because you have no vitamin B3, if you guys remember in the first semester, we talked about if there's no B3, it's going to lead to the 3Ds, similar to pellagra symptoms. Actually, they are pellagra symptoms, which are like dermatitis, dementia, and diarrhea. So here we might notice some rashes. We're gonna notice more, just more notice dermatitis. We're gonna have to notice the patient doesn't have the best memory, and hatta they're gonna have diarrhea. Okay? So let's go with the questions now. So in liver disease, I'm gonna, by the way, just like take 2 seconds and then just show the answer, okay? Because I don't want to take too much time. So in liver disease, the enzymes AST and ALT leak into blood from damaged liver cells. Both of these enzymes have which one of the following in common? The answer is E. They both convert alpha ketoglutarate to glutamate. Okay. I hope this is E. The waste nitrogen from skeletal amino acid catabolism is diverted to the liver for incorporation into urea and which of the following compounds? The answer is A, alanine. Okay. Which of the following represents the major compound of circulation responsible for transport of nitrogen? And the answer here is D, glutamine. Actually, sorry, this is probably wrong. It is glutamate because we talked about like the responsible for the transport of nitrogen is glutamate. But glutamine could work because it contains glutamate as well. I will check with you guys and let you know and after this video, inshallah. Sorry about this. Which of the following vitamins is critically important in the overall process of nitrogen transfer from alpha amino acids to alpha keto acids? And this is C, vitamin B6. Okay. And yeah, that's about it. We talked about it. It's basically a derivative of vitamin B6, which is, your PLP. Oh, and yeah, final question. In this transamination reaction on the right, which one of the following are the products of X and Y? And for those who said B, you are correct. Aspartate and alpha ketoglutarate. So now let's talk now about the urea cycle. Now the urea cycle, we kept talking about it in the first lecture, and trust me, it's easy. Both lectures the first two lectures, in my opinion, are actually easy to understand. They're short and concise. So let's begin. The urea cycle, for starters, it is basically in most tissues except muscles, and ammonia is transported safely in the form of glutamine. Uh, we already discussed this and talked about this, and this is basically done through glutamine synthesis or synthetase, sorry. And glutamine synthetase, I said it carries glutamate and ammonia. So yeah, and yeah, apart from that, here we're gonna have our glutamate, ammonia, and ATP. Through glutamine synthetase, it's gonna eventually form glutamine, and the glutamine, basically the blood, it transported to the liver and the kidney, as you guys remember in the steps we already talked about. If you remember the glucose alanine cycle, it will then eventually to break down and then release glutamate and ammonia at the liver eventually. And then after that, glutamate and NR3, they're gonna go and undergo sorry glutamate's going to undergo oxidative deamination to form alpha ketoglutarate and ammonia. And finally, NH3 is then going to undergo the urea cycle to become urea. Okay, so this is basically the steps of the last lecture, and now we're like, the last three steps, or sorry, the last step is basically this lecture. Okay, by the way, urea, okay, if you ask which amounts is it found primarily highest in, it is found primarily highest in the liver. However, they are excreted through the kidneys, obviously, because it's going to be in the urine. So let's talk about the urea composition and transports. Okay, the urea composition. Okay, here we have this urea as the amino acid. Sorry, not amino acid. The urea itself, the structure. And let's talk about where each composition comes from. For starters, we have here, if you know, you have one carbon, you have two NH2s, and then you have an oxygen behind. Now let's focus on the carbon and the two NH2s. So each of them get their sources, by the way. NH2 here, it gets it from aspartate, okay? And this is mainly from our ASTs, obviously. Uh, our carbon, it comes from carbon dioxide, and this is mainly from the form of bicarbonate. Okay, don't confuse it with the carbon skeleton producing carbon dioxide. It actually comes from the bicarbonate, okay? And finally, NH2 here, it mainly gets it through NH4 plus or ammonia, and this is through oxidative deamination. We're familiar with this. Now, for this to occur, we have two enzymes. Oh, wait, sorry. What acts as also, there's something important here in the steps we're going to talk about. It acts as a shuttle and carrier for intermediates during urea production. This is just a head start for what we're going to talk about, the urea cycle. And it's basically orthonin. Okay, we're going to talk about it now. So here, this is the whole cycle. Don't you guys worry. It might look, packed and you might not understand it well at first. But here, actually, it's a very simple cycle. So we're gonna begin here, almost in our from our orthonin. So our orthonin, okay, is gonna enter the mitochondrial matrix, and within the mitochondrial matrix, we're gonna have two main enzymes, okay, or sorry, we're gonna have like a steps that occur here, okay orthonin and at the mitochondrial matrix, you're gonna have it combined with something carbamoyl phosphate. Now, carbamoyl phosphate, okay, is basically made from carbon dioxide and NH4. Basically, the two byproducts that make our urea, it makes these two first, okay, and they're gonna form eventually called something called the carbamoyl phosphate. And then carbamoyl phosphate is going to combine with orthonin to form citrulline. And basically, this is the first step, and this is the only step that happens in the mitochondrial matrix, okay? So orthonin and carbonyl phosphate eventually make citrulline, and these steps are in the mitochondria. And basically, it's a combination of carbon dioxide and NH3 or NH4 plus. Anyways, after this, we have our second step, which is basically citrulline and aspartate, which is our third composition of our urea. They're going to combine together to form something called arginine and basically, this is like the way I memorize it is Argentina. And succinate, and it's just familiar with us. Um, just remember it as this because it makes a really big molecule called arginosuccinate. Now, arginosuccinate, it cannot remain the same. We need to break it down and cleave it. So we're gonna arginosuccinate is cleaved into eventually two byproducts, which are your fumarate and arginine. Okay. Here I didn't mention fumarate, but no fumarate is actually important. Okay. And finally, our arginine is gonna, combine with water to form urea eventually and orthonin. It's gonna regenerate orthonin so it can begin the whole cycle again. you know? So if I want to have a mnemonic, I learned this from a PAL session, to be honest. It's really good mnemonic. Orange colored cats always ask for awesome umbrellas. Orange for orthenine, carbamoyl phosphate, citrulline, aspartate, and then you have arginine succinate. F is fumarate here. Awesome is Bismillah. After fumarate, we have arginine, and then U is for the urea. So this is the mnemonic I'd memorize and recommend, guys. Sorry about that. Now the next thing here we have is gonna be about sorry, our carbonyl formation. Our carbonyl formation, for starters, it takes along the just citrulline formation, and there are only two steps taken in the mitochondria. The rest is in the cytosol. Okay, so carbonyl phosphate and citrulline synthesis. This is all only in the mitochondria. Okay, and this is a high yield, by the way. They're gonna probably ask about this. And now we need also for this to occur, there are two main enzymes within the mitochondria that help with this synthesis. Okay, one is called شو اسمه carbonyl phosphatase synthetase 1. Okay, and I don't know why they said one. We don't have two or anything. Just remember this carbonyl phosphatase synthetase, and it's just easy carbonyl phosphate synthetase. You can just remember it like that also. And this is basically the most important enzyme because it's the rate limiting enzyme. Okay. And apart from that, it is responsible for carbonyl phosphatase synthesis, you know? Now, another thing that's really important, carbonyl phosphatase, it requires a few things, okay? For starters, it requires 2 ATP. This wasn't mentioned. So remember, it requires 2 ATP. It requires bicarbonate, which, makes our CO2, if you guys remember. And apart from our bicarbonate, it also requires the third thing, which is ammonia. Nice. So these are going to eventually make your carbonyl phosphatase through carbonyl phosphatase synthetase. And they are basically regulated by another enzyme, which is called N-acetylglutamate. So yeah, we have something regulating this step, and this step is going to eventually make it. And we're going to talk about more about this bad guy here because he actually is really important. So here, this is actually mainly regulated basically through produced after a protein meal, and it is also an obligate activator. Okay, and we're talking about N-acetylglutamate here, by the way. It's just produced after protein meal, and it is an obligate activator, meaning it is forced and required to activate. So let's talk about the second enzyme, which is ornithine transcarbamylase. Sorry, the name is a bit hard here, but this concept is very easy. Don't worry. So here it's a bit different, and it's much simpler. Here we're gonna have the carbonyl phosphate, and then here, I'm sorry, you guys can see this is ornithine. It's gonna join with ornithine transcarbamylase, and then eventually it's gonna yield citrulline. Okay, so this is what's responsible for yielding citrulline and facilitates the reaction and the production and release citrulline into the cytosol. Okay. And it's also facilitated by the rupture of phosphoanhydride bonds. So we have a phosphoanhydride bond, and it ruptures it to eventually give inorganic phosphate. And yeah, because we need something, high in energy to act upon this, which is why all of this occurs in the mitochondria eventually. So let's talk about the other thing. Let's talk about the other enzymes responsible within the steps, like the steps after. So if you guys recognize, after that, we have citrulline, right? Citrulline is going to combine with something which is our aspartate, right? Citrulline is going to combine with our aspartate, and it's going to use the enzyme argininosuccinate synthetase to make argininosuccinate. Okay. And basically, yeah, this is as simple as it is. It catalyzes condensation of citrulline to form argininosuccinate. The second enzyme we have here would be argininosuccinase. Okay, so basically A is just to break argininosuccinate, because I told you guys this is a big molecule. We need to break it down into arginine and fumarate, right? And yeah, that's about it. It retains the carbon skeleton as aspartate as a fumarate molecule. Okay, so basically it's saying, the aspartate, the carbon skeleton will eventually become fumarate eventually, if you guys remember, after the reaction occurs, right? And then we have arginine is gonna become orthonine and urea through arginase. And yeah, basically catalyzes this. And yeah, this is the rest of the steps. And this is a very important concept, okay? Here, we're using 3 ATP molecules, but 4 high energy phosphate bonds. So we're not really just using,
three ATP. We're using four high energy phosphate bonds, and because this is all coming together, okay? And basically, you have two ATP and two high energy phosphate bonds. They're used in the synthesis of carbamoyl and phosphate. And then you have one ATP and two high energy phosphate bonds, and this is used an ATP to convert to AMP in the synthesis of argininosuccinate. Okay, so basically, just to emphasize, here it's using one ATP, two high energy bonds. Here it's using two ATP. Okay, it's using two ATP to synthesize carbamoyl phosphate, if you remember, right? And because of this, both of them have two high energy bonds, and the one ATP here it has actually also two high energy bonds. And yeah, so we have three ATP and four high energy bonds. So this is how I would remember it. You have four high energy phosphate bonds, but in fact, you have three ATP being used. Okay, and this is very high yield. She talked about a lot about this. She's gonna probably get this. Let's talk about the linkage of cycles. Let's link the cycles together, right? And I'm not talking about the year cycle. I'm talking year cycle of other cycles. You know, like what? Yeah. So here, when we form the carbon monophosphate and then the citrulline, we're gonna form eventually the arginine and succinate, and then it's gonna be cleaved because it's a big molecule into arginine fumarate. So fumarate is actually gonna enter another cycle, and it connects it with, as you guys remember, called the citric acid cycle. Also, the TCA cycle, basically, and it links it so it can, eventually form malate. And after fumarate forms malate, malate is going to form oxaloacetate. And here's oxaloacetate. If you remember, guys, the reaction with the alpha amino acid, it's going to form aspartate and alpha keto acid. And شو اسمه? Now, this is like our AST here, right, as the enzyme. And aspartate is regenerated, so it can rejoin its citrulline to form arginine. So it's just, continuing to use. And then here you have urea and orthonine, obviously, you know. And yeah, that's basically we have fumarate forming eventually malate. Malate is going to form oxaloacetate. And then oxaloacetate is transmitted to the aspartate, and the aspartate carries amino groups from the other amino acids into the urea cycle. So yeah, this is why I'm telling you guys, this is very important. It is mainly in the synthesis of, as I told you guys, if you remember, mainly it acts as glutamate and oxaloacetate to synthesize aspartate and alpha ketoglutarate, not the opposite, you know. And alanine, it forms glutamate mainly and pyruvate. However, here it works in the opposite, you know. So yeah, that's about it. And this is again through ASTs. Please remember this, okay? So another thing, the overall reaction, okay, and I want you guys to remember this, okay? And there's an important concept. We have the overall reaction, which is our two NH3s, right? We use two NH3s, one from ammonia and one from aspartate, right? We have the CO2, we have three ATPs, as I told you guys, and eventually it's gonna make urea, two ADPs, an AMP, and شو اسمه P pi and 2 pi. Now you're like, wait, what are these? Uh, P pi to 2 pi, this is mainly done by pyrophosphates. Basically, you have a pyrophosphate. And yeah, that's all you need to know. Yeah, that's that. Now, regulation of the urea cycle. Let's talk about how awesome the urea cycle is regulated. So, we said, مثلا when we have a high protein meal, our N-acetylglutamine, شو اسمه, it's in high levels, right? So here, the urea cycle is also elevated or regulated through. For starters, it's regulated by the levels of enzymes. It's also, by diet and food intake. Sorry, not intake. Okay. And finally, the concentration of cycle intermediates. Okay. So yeah, that's about it. The more like, intermediates you have, the more like, the urea cycle is going to need to be active, the levels of enzymes, if you have a high activity, obviously, and finally the diet and food, if you're having, a lot of protein, we need to degrade this protein to, and more like NH3 is going to enter the urea cycle, you know? So yeah, that's about it. And there are some intermediates that can be converted to orthogonine, sorry. And this is going to become arginine from our diet, okay? And glutamate from intestinal enzymes. Just remember, intermediates that can be converted to orthogonine immediately, right? So we can continue. It's basically arginine from diet. It's basically we have like a high arginine, diet. We're gonna have a higher orthogonine concentration. It's gonna make more of the urea cycle. And then here, glutamate from intestinal enzymes. That's about it. So another important thing here I want you guys to focus on, even though the cycle is right spontaneous, right? Okay, like spontaneous and it keeps going because we're synced to the citric acid cycle, right? It has a fine control and it's mainly controlled by something important, also called your carbamoyl phosphatase synthetase. Okay, and basically just know that the main control is through the carbamoyl phosphatase synthetase, because technically it is the first step, right? It's the rate limiting step. And because of that, if we don't have carbon phosphatase, it cannot combine with orthonine to form citrulline, you know? So think of it that way. It is the most important like step. We talked about it and we talked about what it's regulated by, which was N-acetylglutamate. And it is basically made from acetyl-CoA and glutamate in the liver. Okay. And you know how we talked about how N-acetylglutamate regulates carbon phosphatase synthesis? It actually regulates, it is regulated by also arginine. So yeah, this is why arginine in diet can be, شو اسمه, can be, important because it's going to convert to orthonine and it's gonna regulate an acetyl glutamate. So yeah, I hope that's like a fun summary, a fun fact you just learned. And yeah, the summary basically, an acetyl glutamate is mainly it's just more regulated through acetyl CoA. I didn't mention this by the way. It's regulated by acetyl CoA, glutamate, and arginine. You know, and yeah, that's about it. So let's get into the enzyme deficiencies. We're almost near the end of this lecture. A very easy lecture, don't worry. And from, the enzyme deficiencies, we can have some enzyme deficiencies, and all of them are bad, but there are some specific ones that can be bad. I'll talk about them. So we have all of these enzymes, as you guys can recognize them, arginase, and acetylglutamate synthetase. By the way, this is what makes your N-acetylglutamate synthetase regulated by our arginine, as we talked about in the last one, or in the last slide. Okay. The enzyme deficiencies that we want to mainly talk about here are carbamoyl phosphatase synthetase 1 and orthonine transcarbamylase. Now, why is this important? Because both of them are actually very similar, but they differ in some things. Okay, so here I made this table, and to be honest, yes, I did use GPT, but it really explains it well, right? So for starters, here, the biochemical findings of having deficiencies in both of these, you're gonna notice increased NH4 plus, and which is hyperammonemia, right? Because we have a lot of ammonia and it's not being converted, to just what we need. Here we're gonna also have increased NH4 plus and hyperammonemia, even though we're converting it to carbamoyl phosphatase. Carbamoyl phosphatase is still retaining NH4 plus, which is why we have increased NH4 plus, even though we're using it to generate carbamoyl phosphate, right? So in our blood findings, we're gonna actually notice a very high blood glutamine levels. Okay, and both of them are you're just gonna notice high blood glutamine. Okay, the kidney findings, because also they're dysfunctional. There's low urea to begin with. Okay, and because of that, all of this there's this impaired urea cycle. However, here this is the most important part I want you guys to focus on. Okay, the neurological findings here is that here you have no erotic aciduria, and I'm going to talk about it in a second. Here, there is erotic aciduria. Basically, here you carbonyl phosphate, you have high NH4, high CO2. You're not converting it. Here you have شو اسمه carbamoyl phosphate, and it retains NH4 plus. But the thing here, in orthonine transcarbamylase, there's something important that's produced, and it's basically oh wait, let's talk about the other things first. I'll explain this part here, I promise. So here we have autosomal recessive, it's X-linked recessive, and here basically to treat it, you have to have a low protein diet for both of them because low protein doesn't stimulate urea. So this is what I want to talk about. Orotic acid and uracil are intermediates in the pyrimidine. The increase in orthonine transcarbamylase deficiency, orotic acid and uracil, basically they're gonna cause aciduria or orotic aciduria due to their increased amounts. They're not found in carbamoyl phosphatase synthetase. They're only found in orthonine transcarbamylase, which is why when you have high amounts of these, it's gonna cause orotic aciduria. Okay, you don't need to know what orotic acid, or you obviously know what uracil is, but you don't really need to know what orotic acid is. And yeah, apart from that, symptoms will involve lethargy, vomiting, cerebral edema, etc. Because these all, we're gonna definitely have like neurological problems, and we're gonna have vomiting and lethargy, you know. And yeah, that's about it. Increased glycine, glutamate, and alanine is noticed because you have an impaired urea cycle, and it's not being able to control. And there's also increased NH4 plus for that reason. Now, how do we treat if we're having increased glycine and glutamine? This is bad our body. So if you want to treat the patient, obviously, apart from a low protein diet, we need to treat for their excess glycine and glutamine. And this is done by two things that are very important. Glycine, yeah, to treat excess byproducts, you know. Glycine here, okay, is treated by benzoate, okay, and benzoate is just going to convert it to a state called hyperate, so you don't have an excess amount, okay. Glutamine, on the other hand, okay, is gonna be treated by phenyl acetate, and phenyl acetate is gonna eventually form phenyl acetylglutamine. I just wrote GLM, so you know, because I didn't already have space. But yeah, that's about it. That's all you need to understand here in the slide. Just know the benzoate phenyl acetate. Remember that no erotic aciduria. Here, the symptoms, remember them. And yeah, that's about it. The other enzymes that are obviously they're going to cause problems, but these are the most severe. And by the way, fun fact, arginine transcarbamylase is actually the most common one to be deficient out of all of these. You know? So, enzyme deficiencies, finally, let's talk about the other stuff. So, here we have obviously, we talked about the argininosuccinate, right? But we want to talk about the argininosuccinase deficiency. If we don't have argininosuccinase deficiency, if we have a problem with it, obviously we're not gonna have fumarate and arginine. We're gonna retain argininosuccinate as like, which is a big molecule, right? And here the treatment, we said we have a low protein diet in arginine. Okay. So yeah, basically here we have the treatment. And as a result, arginine succinate is basically secreted in place of urea. And this is, you know, bad, but just small. This is the way to treat it and all that. And let's remember that ammonia is elevated in شو اسمه CPS deficiency, while orotic acid NH3 and CP is elevated in OTC deficiency. Here, basically, it's carbonyl phosphatase in OTC, which is our شو اسمه بسم الله الرحمن الرحيم. It was our orthorhine transcarbamylase deficiency. Here, only ammonia is elevated in شو اسمه carbonyl phosphatase synthetase deficiency of an enzyme. Okay. And yeah, here I included it from the slide so you guys can get it immediately. NH4 toxicity may be due to excess glutamine. High glutamine levels are found in cerebrospinal fluid. Alpha ketoglutarate and NH4 form glutamate, and NH4 will eventually form glutamine, which is why we have excess glutamine that we talked about. High glutamine and glutamate can lead to brain damage, you know, by producing osmotic effects, which is why we talked about cerebral edema as a symptom, right? So yeah, that's about it. Let's solve some questions. A newborn that developed seizures 2 days after birth was diagnosed with elevated serum NH4 plus, glutamate, and alanine. A neonatal urea cycle defect is suspected, so a urinalysis was performed to assay for orotic acid and serum assayed for citrulline. The levels of both were found not to be elevated. A defect in which the enzymes in the urea cycle is more likely to account for the symptoms on birth in this infant. The answer here is carbonyl phosphate synthetase, which is D basically. Basically, if we realize we already have increased bismillah, we have increased NH4 plus, glutamate and alanine, but we're not noticing like erotic acid. Like basically here, it's telling you the levels were both not to be elevated. You know, so this is immediately D because we're having high NH4 glutamine and alanine, or glutamate and alanine. So here, a 2-day-old infant is brought to the ER suffering a seizure. Analysis from serum NH4 plus reveals it to be quite high. Suspecting a urecycle impairment, the ER physician initiates hemodialysis, which reduces the NH4 plus levels. Subsequent to the hemodialysis and treated with supplemental arginine, which maintains the reduction in serum NH4. The ability of arginine to render this effect stems from its role in activating the synthesis of an activator of one of the enzymes of the urea cycle. Which of the following represents the potent activator whose synthesis is induced by arginine? If you guys remember, we talked about arginine being a regulator, and the answer here would be E, because arginine regulates N-acetylglutamate, and yeah. Which one of the following occurs in the urea cycle? Very easy question. The answer here is D, arginine directly reacts with carbon monophosphate to form citrulline. Which enzymes are responsible for reducing the direct donors of nitrogen into the pathway producing urea? Finally, answer here is E, aspartate amine transferase and carbon monophosphatase synthetase. So yeah, let's talk on. Yeah, finally, the last question here. A 3-week-old infant has been having the intermittent vomiting and convulsions. She also has had episodes of screaming and hyperventilation. The infant has been lethargic between episodes, thus revealing an expanded abdomen. Blood values show decreased citrulline amounts as well as decreased BUN. Decreased BUN, by the way, it's blood urine nitrogen. Okay, so don't get confused. What other clinical outcomes would be expected in this infant? If you said the answer is C, you are correct. Increased blood glutamine, increased orotic acid because it's ornithine transcarbamylase deficiency. So now let's begin talking about amino acid degradation generally. Now, this is our lecture that this is like one of the harder lectures moving on, but inshallah it's going to be easy. So obviously we want to talk about first the site and fate of amino acid degradation. So where does sorry, where does amino acid degradation occur also? It mainly occurs in the liver, okay? And like the urea cycle, but it also can occur in the kidneys, the muscles, and all of that, okay? So here, basically, we have all of this, okay? And because of that, there are some exceptions, by the way. Not all amino acids are degraded in these regions. You have, if you remember, guys, branched amino acids, which were leucine, isoleucine, and valine. Their degradation takes place in skeletal muscles. Okay, so here you have the chains are broken down or degraded in skeletal muscles, and yeah, this is important. Please recognize this because this was asked about before at TBL. So here we have the amino acid deamination. The removal of an amino group. Here you're gonna have the amino group. It's gonna be incorporated into other compounds like urea or excreted, right? Here, the after we move the amine group, we are left with the just alpha keto acid, also your carbon skeleton. This carbon skeleton is either going to be catabolized for energy, or it's going to be synthesized into other compounds like carbon dioxide, ketone bodies, everything we already talked about. So carboskeleton, also called alpha keto acid, which remains after deamination, is either synthesized in other compounds or catabolized for energy. The catabolized products may be okay. Yeah. So the products of the carbon skeleton, catabolizing it, might produce products of two types. The catabolized product may be ketogenic, or glucogenic, and we're going to talk about them. Okay. Now, like how we talked about the catabolized products that are going to be produced by the carbon skeleton, as we talked about it, amino acids are also they may be classified into ketogenic or glucogenic because technically, if we're breaking down the carbon skeleton, this may like contribute to the formation of other amino acids, and the amino acids themselves, Aslan, before they're deaminated, they may be ketogenic or glucogenic, contributing to the products that are gonna be either ketogenic or glucogenic or even both. Okay. So yeah, and also something very important, leucine and lysine are the only amino acids that are ketogenic. Okay, just keep this in mind because we're gonna talk about other stuff. But just remember, LL, leucine, lysine, both of them are ketogenic. Now for amino acid catabolism, okay, here we're gonna have to understand this picture first of all. So we talked about this removal of amino group, metabolism of these stuff, and it's eventually gonna produce the 7 products, okay, which is either the ketogenic or glucogenic as we discussed. The products now we're gonna talk about, you have ketogenic, which is acetyl-CoA, while here is acetyl-acetyl-CoA. So the mnemonic I use for memorizing the 5 here, right? While here there are two A's that begin with acetyl-acetyl or acetyl, so they're similar, you might recognize them. But here to remember these, I use a mnemonic called POFAS. POFAS here, pyruvate, oxaloacetate, fumarate, alpha ketoglutarate, and succinyl-CoA. Now, just small. Eventually, the ketogenic, by the way, they're going to provide lipids and energy, while glucogenic is going to provide lipids, energy, and glucose. Now, the reason glucose is because we're going to talk about them here. Pyruvate can, you don't need to know what I'm about to show you guys, but it will make so much sense when you understand why it makes glucose. So pyruvate will enter gluconeogenesis to make new glucose. Oxaloacetate will also enter gluconeogenesis to make new glucose. However, the last 3 fumarate, alpha ketoglutarate, and succinyl-CoA, they will all be converted into oxaloacetate to be eventually, to enter gluconeogenesis. And yeah, in the Krebs cycle. So what about the acetyl-CoA and acetyl-acetyl-CoA? What are their roles? This is very simple. If you studied the other lectures, previous lectures, like fatty acid oxidation, you remember that acetyl-CoA is part of the ketone body synthesis or fatty acid synthesis, and so is acetyl-acetyl-CoA. Sorry about that. And yeah. So what? Let's continue to talk about the catabolism. Now, as we said, we can as I told you guys in the other slide, we can have products that are either ketogenic, glucogenic, or even both. Let's talk about amino acids that can be ketogenic and glucogenic. Okay, so this can happen and it can occur basically when ketogenic and glucogenic byproducts as a result. So in the amino acid, if it breaks down, like if we take one amino acid and we catabolize it, and we notice, like for example, you have acetyl CoA and fumarate, right? We're gonna notice that this enzyme is technically just ketogenic and glucogenic because it has also byproducts of both, صح? And here there are five amino acids that are ketogenic and glucogenic, and I made a really good mnemonic for this one. For those who love to watch The Pit, okay. Here it's the Pit with three T's, okay. P for phenylalanine, and broken down to fumarate and acetate. Isoleucine, which is broken down to acetyl CoA and succinyl CoA. T is for fumarate and it's broken down to fumarate and acetate. So we're noticing that this is acetoacetate or acetyl CoA, and here's fumarate from the other one. شو اسمه here it makes acetyl CoA and succinyl CoA. You have threonine. Threonine here is we don't really need to know them, and tryptophan we don't really need to know. So if I were to tell you guys to memorize all of these, I would just be giving you guys a headache, which is why I want to tell you guys just memorize phenylalanine and tyrosine. Both of them make fumarate and acetate, right? And the reason they're very important is we're going to talk about them in the later lectures, like phenylalanine and tyrosine metabolism. But we'll get to that when we need to, okay? So the essential amino acids, we've actually taken this in the first semester. Very easy. So essential amino acids, basically all 20 amino acids are required for protein synthesis, and we may be able to obtain them through, again, dietary sources, de novo synthesis, and protein breakdown. We already discussed this in the first lecture. Let's understand, however, essential amino acids. So basically, essential amino acids from these 20 amino acids, some essential amino acids cannot be synthesized in adequate amounts or also in whole, right? And therefore, we must obtain them from diet, right? Which is why we always tell you, go eat your protein for the gym enthusiasts, because there are some amino acids we need, right? And here, arginine is conditionally also essential, is particularly during periods of growth. Just recognize this because it is a very important amino acid for, regulating also N-acetylglutamine in the other lecture. So here, let's talk about the essential amino acid mnemonic, the very famous mnemonic we all love, PVT Tim Hall. Just go over it again. I'm sure you guys, remember it still, and it's still somewhere deep in our, cortex. So here, don't get overwhelmed. This is nothing to memorize. The major pathways of amino acid degradation, we're going to talk about the seven of these degradation, and then in later lectures, we're going to break down and understand each of these, right? So first of all, you have the removal of amino group and blah, blah, blah. You guys are going to see what I'm talking about because this is like the point where we want to discuss all the lectures later on. So the first thing is from the objectives we have is the removal of the amino group. The removal of the amino group, okay? Like some amino acids can be useful upon the removal of the amino groups, where they contribute to Krebs cycle, as we talked about how they enter gluconeogenesis for glucose synthesis, right? And these amino acids, an example of them are alanine, which contributes to pyruvate through ALTs, aspartate to oxaloacetate through ASCs, we already know this, and hatta glutamate to alpha ketoglutarate via aminotransferases. We already know this. نحن. It's very easy, صح? But there are two new ones I want you guys to focus on. Asparagine, which makes aspartate through via asparaginase. Okay. And apart from that, you have glutamine, which makes glutamate through glutaminase. Okay. They're very easy because you can just see that enzyme is just saying it's breaking this into this and this into this through, their name. And they're to be recycled in the steps above, to eventually form the byproducts for Krebs cycle, you know? So yeah. So apart from that, another important thing I want you guys to recognize is that these enzymes, they are mainly facilitated by transaminases and deamination by using specific enzymes, as we talked about, aminotransferases, asparaginase, blah, blah, blah. So what is very important here? The most important one from all of these is asparagine to aspartate or asparaginase. Why? This is actually related to a clinical concept we're going to talk about. So in things like بعيد الشر, we have leukemia and lymphoma, right? Our leukemia and lymphoma, we actually lack asparaginase as an enzyme in it. And the reason it does this is because it uses asparagine as it's like an energy source or like growth source. That's why the tumors can grow in lymphoma and all that, right? And for that reason, uh, to help patients with lymphoma and leukemia, we need to give them asparaginase to break down and not allow the tumors to have asparagine as their growth source and to eventually form aspartate, which will contribute to, aspartate to oxaloacetate through just aspartate aminotransferase. So yeah, this is just basically what I said. Yeah, deprives the tumor cells of their source. So let's talk about the second thing on the checklist there, our phenylalanine and tyrosine metabolism. Okay, so our phenylalanine and tyrosine metabolism here, phenylalanine, just to understand it, it is basically tyrosine is basically a hydroxylated version of phenylalanine. Okay, so just recognize this, and tyrosine is just a hydroxylated version of phenylalanine. And apart from that, how is it converted, Tayyib? There's a reaction for this. Phenylalanine undergoes hydroxylation to tyrosine by phenylalanine hydroxylase and NH4. It uses NH4, but mainly focus on the enzyme phenylalanine and hydroxylase to give a hydroxylated version of it to make tyrosine. Apart from that, if phenylalanine is non-functional, PKU will occur, causing accumulation of phenylalanine. We're gonna get this into later, into lecture 21. I just wanted to give you guys a heads up. If we don't have this enzyme is non-functional, increase of this, decrease of this, and eventually you're gonna end up with the PKU, as you guys have heard from it before in other lectures. Now, why is tyrosine also very important? Tyrosine is very important generally because tyrosine contributes to the formation of other stuff like catecholamines, melanin, and thyroid hormone. This is all very important because we already talked about it. Catecholamines, epinephrine, norepinephrine. This is all talked about in ERP. Now, again, I want you guys to remember that phenylalanine and tyrosine contribute to Krebs cycle through fumarate and acetate when you catabolize them. Let's talk about metabolism. Now, the third thing, the metabolism of threonine, serine, and glycine. You know, now why is this very important? Threonine, serine, and glycine are all related and have the same metabolic pathway. So basically here, serine itself, along with threonine, is eventually going to contribute to the formation of glycine. Now, شو اسمه? And the way it does this, serine, okay, it will lose a carbon molecule, but threonine, it loses an acetyl-CoA, okay? And when it loses an acetyl-CoA, it will eventually form glycine, and glycine will then end up either شو اسمه, forming CO2 and NH3, or it will form glyoxylate, which will form oxalate later on. And we're going to talk about these importantly. So as we said here, threonine loses acetyl-CoA, serine loses a carbon molecule, and glycine will end up either forming CO2, NH3, and also something very important called methylene. Okay, and this is the major pathway, by the way, it takes. Just imagine it going down. Here, glycine, the minor pathway will form glyoxylate, but we'll talk more about this. So to form CO2, NH3, and methylene, it requires three things. Okay, and I want you guys to count with me on your fingers. First of all, it requires glycine cleavage enzyme, it requires B6, and it requires THF by using one carbon molecule for the THF methylene. Okay, that's about it. And yeah, here basically it is. It uses B6, it uses a glycine cleavage enzyme to eventually form CO2 NH4, and it will also, use a H4 folate to give an methylene H4 folate, you know? And now to form glyoxylate and oxalate, which is the minor, why is this very important? If we have a lot of glycine levels in our body, and we talked about why we need to remove glycine and glutamine, right? If you remember benzoate and all that, like, during the deficiencies of the enzymes and the urea cycle, why do we need to remove glycine? Glycine, it will contribute to the formation of glyoxylate if we have too much of it, right? Because we're gonna, take this is the major pathway. So if you have too much of this, there's gonna be too much of here, CO2 and NH3, right? But there will also be too much glyoxylate. Glyoxylate will form oxalate, and the reason this is not as good is because it might combine with calcium, and when it combines with calcium, it will eventually contribute to forming calcium oxalate stones, and which, if you guys recognize them, these are kidney stones, which is why if we have too much glycine, it might contribute to kidney stones or calcium oxalate kidney stones if too much glycine. So yeah, that's about it. Let's talk about the fourth thing on the checklist, methionine and cysteine metabolism. So what's important about this, and I did a whole step to break it down, so inshallah it's easy. So for starters, methionine and cysteine, this is by the way the most important one. This is so high yield, and it's high yield in other lectures. We're gonna talk about it. Methionine, we're gonna take it, okay? Methionine is the one undergo it will form eventually something called SAM or SAM, also called S-adenosylmethionine. And the way it does this, it occurs by a step where we're going to use ATP. So methionine plus ATP forms SAM, S-adenosylmethionine, right? You can see like where like it gets the name because there are factors of methionine and ATP. And this is required by an enzyme called methionine adenosyl transferase. Not as high yield, but still remember it, okay? Because methionine it transfers the adenosyl to form adenosyl شو اسمه methionine. And then afterwards, its SAM is then going to donate a methyl group from here. So it's going to donate a methyl group from methionine, and then it's eventually going to form something called SAH or SAH, that's in Arabic, which is S-adenosyl homocysteine. So S-adenosyl homocysteine is very important because it will contribute to other things, but I just really need to you guys recognize that SAM is the major methyl group donor, okay? And we'll do it through something called methyl transferase, obviously. Yeah, like adenosyl transferase, methyl transferase. So SAH, S-adenosyl homocysteine, because it's a big molecule, it will eventually give two things. We're going to hydrolyze it by adding water and cleave it, and it's eventually going to have homocysteine, and S-adenosine is just going to give adenosine, the fancy way of saying S-adenosine or adenosine. And this is very important, okay, because eventually homocysteine will undergo changes, and these changes will either be, and the reason it needs to undergo changes is because this is toxic. If it remains too much in the body, it may cause some problems, okay? And this is not really a good molecule, which is why we have two pathways to ensure it's like, removed. And this is by through two things, okay? This is either by remethylation into methionine. So remethylation, it requires three factors, and this is very important. Wait, I don't know if you guys can remove it. Okay, yeah, can you see it now? Oh, wait. Okay, there. So it requires B12, folate, and THF to form methionine. And basically, yeah, it's just regenerating methionine over so it can continue, And it requires B12, folate, and THF. Again, okay? Please remember this. And apart from remethylation, it might do a step called transsulfuration into cysteine. So basically transsulfuration because it uses B6. So here I imagine because it needs to generate methionine, it needs to use a higher amount of vitamin. So it uses vitamin B12, a higher amount, which is B12. While here, because I imagine it's gonna continue the cycle, it's not gonna use as big, so it uses B6. That's how maybe I remember it. I hope this helps. And eventually, after that, we have cysteine. We already talked about methionine metabolism. But now let's talk about cysteine metabolism. Cysteine will eventually be catabolized, right? And it will eventually form pyruvate and sulfite. Okay? And yeah, this it's undergoing catabolism. And apart from that, pyruvate and sulfite will eventually, okay, sulfite here, sorry, not pyruvate, sulfite, it's a molecule that we need to remove, right? So to remove it, we need to oxidate it to form sulfate, okay? And it's going to be excreted in urine, okay? And yeah, that's the whole cycle. It's very important, okay? Please, like repeat this video as much times as you can, but just mainly focus on this point exactly. And yeah, now let's talk about the key points of the entire step. The key points for starters. First of all, we said S-adenosylmethionine is also short for SAM, is the major methyl donator for RNA and DNA and other stuff, right? Also for to donate it and to give S-adenosyl homocysteine. Second thing is upon hydration of S-adenosyl homocysteine, not only homocysteine generated, but also adenosine from adenosyl part. But it's not really focused on. What's gonna then undergo is homocysteine because it needs to, it's toxic material and it needs to be removed and regenerated into other stuff. And the third and final thing, this is the more important one. Now, we talked about how it undergoes transsulfuration from homocysteine. There's actually another pathway cysteine is formed, okay? And I just wrote it in text because it is unfortunately high yield, but I couldn't imagine how I would generate it. So I just talked about it here. it exists another pathway for cysteine through transsulfuration. There's another transsulfuration that occurs and basically involves the carbon skeleton of serine and the sulfur of methionine. So if you were to take methionine, its carbon skeleton, and when you take the carbon skeleton of methionine and take this شو اسمه sorry, the carbon skeleton from serine and the sulfur from methionine and add both of them, you can just immediately form cysteine instead of doing the whole process here. So yeah, that's it. Now let's talk about the fifth the checklist, which is histidine metabolism. Histidine metabolism is one of the easier ones, so don't worry, and it's not as high yield, but if you understand it, you're good to go. So histidine, it's basically going to be converted into urocanic acid, and basically it is by the enzyme histidase. And when it does this, it will release NH4 plus. Urocanic acid will eventually form something called N-formimino-glutamate, also called FIGLU. Now, just memorize it as FIGLU or IGLU, I guess. And FIGLU will be converted to glutamate through something called tetrahydrofolate. And if you guys remember in the last the last slide, we said THF, folate, and B12 are important for remethylation of homocysteine into methionine. صح? Well, would you want to know that THF is actually a precursor of شو اسمه, it's directly derived from folate and B12. That's why I put it with them there, because technically THF contributes to formation of methionine from homocysteine, because folate and B12, are basically just THF is basically a precursor of both of them. Okay. And also glutamate eventually forms alpha ketoglutarate. This is a step that's supposed to be easy, and we already know this, right? So yeah, THF is directly derived from folate and vitamin B12. It explains why FIGLU accumulates and presents in urine when during folate deficiency. If we have folate deficiency, we don't have obviously we don't have vitamin B12, and because we don't have both of these, we don't have THF, and THF is just going to cause FIGLU accumulation in urine, during folate deficiency. THF, okay, is responsible for other stuff. This is not as high yield, but it is just good to understand. THF is responsible for other things like N-methyl THF. This is actually the most important one here. You just need to know it. It helps with the remethylation of homocysteine to methionine. So just know this, as we talked about it before. N-formyl THF, formation of urine. N-methyl THF, DMP to DTMP. You don't really need to know these. Just know N-methyl THF because we already know it remethylates homocysteine to methionine because homocysteine is toxic, right? Yeah, and also a deficiency in folate can cause deficiency in THF, which can also cause megaloblastic anemia. Okay, so yeah, if they were to ask what disease is likely from deficiency of folate, megaloblastic anemia is basically the answer. We're almost done, guys, with the lecture. I'm sorry this lecture is long. It is actually, unfortunately, one of the longer lectures here. So branch chain amino acid degradation. Let's talk about the sixth thing on the lecture, the checkpoints or the checklist. Let us remember by remembering our branched chain amino acids. We already talked about them. The branched chain amino acids are degraded where in skeletal muscles. And because they're degraded in skeletal muscles, you have three: isoleucine, leucine, and valine, right? And there are actually, because we have these, we actually have enzymes, or sorry, the main enzyme here that supports, the amino acid degradation is called branched chain keto acid dehydrogenase. Basically, it does this by removing a hydrogen and breaking it down further, making it unstable to be broken down. So yeah, that's about it. Now, for every enzyme, we have its coenzyme or cofactors, right? And the coenzymes here, okay, if you guys remember the شو اسمه the mnemonic, the lovely coenzymes for nerds, okay? These are the enzymes that are responsible here. Thymine pyrophosphate, B1, lipoic acid, coenzyme A, vitamin B5, FADH2, vitamin B2, NADH, vitamin B3. And you can remember these as also niacin and I forgot the other one's name, but yeah. This is the mnemonic, and these are supporting branched chain keto acid dehydrogenase. A mnemonic I made, to be honest, in first semester, and a lot of people actually started using it. It was the loud cows from New Zealand as well. You may use this if you ever forget, the first one. And yeah, this is just basically it. And yeah, or you can just use the lovely coenzymes for nerds, or loud cows from New Zealand. So let's talk about the final thing on the checklist here and the final thing of this lecture, which is propionyl-CoA pathway for amino acid metabolism. Now, propionyl-CoA pathway is a very easy pathway where it was discussed in fatty acid oxidation or fatty acid, metabolism and all of that, and the other lectures. And so there are those who studied it. you're going to find this easy. So the amino acids involved in the propionyl CoA formation, to form propionyl CoA, there are four amino acids involved that are going to be eventually degraded, right? They're methionine, valine, isoleucine, and threonine. So I don't really have a mnemonic for this. Maybe you could say MIT is very good, very V valine. So I just made it on the spot. But to be honest, my personal mnemonic is for those who play Valorant. Personal Valorant main is Tejo. Valine, methionine, isoleucine, threonine all contribute to propionyl CoA, which is P, right? So yeah, maybe that's easier for some of you to memorize it. And yeah, now let's talk about the actual steps. So here, for those who studied fatty acids and cholesterol, for schismal, oxidation, all of that, and the other lectures, you're going to notice that we know that it eventually forms propionyl CoA, but we also now know from this lecture that it forms methionine, threonine, isoleucine, and valine. So here you're going to have something called form your propionyl CoA, and your propionyl CoA, which is three carbons, is going to eventually undergo propionyl CoA carboxylase. So we're going to get an extra carbon from propionyl CoA carboxylase, and it will form methyl malonyl CoA, which has four carbons now. Methyl malonyl CoA is gonna have an enzyme called methylmalonyl-CoA mutase, and it's just going to mutate it in a different thing called succinyl-CoA. Succinyl-CoA will either go to citric acid cycle or gluconeogenesis. Now, the more important part I want you guys to recognize here is the enzymes and its cofactors. These are so high yield. Please memorize them, please. Allah khalikum, because this is so high yield. Propionyl-CoA carboxylase, it actually uses biotin or B7 as a cofactor here, while methylmalonyl-CoA mutase uses vitamin B12. Okay, so I'd memorize them that 7, 12, 7, 12, like it goes in order. Okay, so yeah, recognize the enzymes, blah blah blah, vitamin B7 dependent, da, 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 and yeah. Therefore, vitamin B12 deficiency will cause methylmalonic acid accumulation, which is a neurological abnormality because you have a lot of شو اسمه you have a lot of methylmalonyl CoA, and causes basically neurological abnormalities. Let's now get to the questions, okay? Which of the following enzymes is used as a chemotherapeutic agent to treat lymphoblastic leukemia? For those who said C, you are correct. Asparaginase, because it causes asparagine to form into aspartate, so it makes asparagine not available for a growth source or something they can use to grow, which are your tumor cells and leukemia. Which vitamin is necessary for the remethylation of homocysteine to methionine? Vitamin D, vitamin B12, and also there's folate. And apart from folate, oh wait, I think this question might be wrong because I think folate is شو اسمه بسم الله. I think here this is also right. C is also right because vitamin B12 and folate, which is vitamin B9, both contribute to it. I guess this was a mistake in the past questions. But yeah, both of these are correct. But imagine if this was to be B1, for example, vitamin B12 would be the answer here. Okay. Which two amino acids are exclusively ketogenic? You remember, guys, leucine, lysine. A. Sorry. Yeah. PKU results from a deficiency in which enzyme? I mentioned this, and it's going to be talked about more later if you don't get it right. It's okay. B. Phenylalanine hydroxylase. Easy. We also have other things, or the other questions here. Which of the following vitamin-derived cofactors required by the branched chain alpha ketoacid dehydrogenase complex? If you said شو اسمه C, you are correct. Thiamine pyrophosphate, which is B1. Uh, and yeah, that's about it. That's everything you need to know. And now let's talk about this. So as I told you guys here, this lecture speaks about the last lecture, but now it's going to speak about it in more details and more clinical scenarios. Okay, so let's begin. So to start this, phenylalanine and tyrosine are very, very, very important because again, they contribute to the formation of fumarate and acetate. Please don't forget these. These are important because they contribute to Krebs cycle and they're important. Now, we said that phenylalanine, contributes to the formation of tyrosine. Tyrosine will contribute to the formation of dopamine, norepinephrine, epinephrine, melanin, and thyroid hormone. Maybe the new one here is dopamine. I didn't mention the last lecture, but please know that dopamine is also contributed to, okay? So here, the metabolic disorders that we really need to focus on that are going to be included in this lecture are PKU, which we already talked about. We're going to speak about it more, which is phenylketonuria, albinism, because you're going to realize that it has to do something with melanin, and finally, alkaptonuria, which is going to have to do also with the tyrosine. Okay. Now let's talk about something very important. So guys, as you guys see, phenylalanine is basically just phenyl plus alanine. That's it. Okay. I just want you guys to recognize this because there was apparently a past and some people got it wrong. I don't know why, but yeah, there was a past question about this, and just recognize this. Okay. It's mainly made of phenyl and alanine. Okay. So let's talk about the steps here, how it forms tyrosine in more details. Because we said last time, phenylalanine hydroxylase forms phenylalanine and forms tyrosine eventually, right? Now, the important part here and the new part here, okay, is actually going to be something called BH4. BH4 is going to be used and it's going to eventually form BH2. Here, we're going to also explain how BH2 is going to form BH4 regenerate and form tyrosine, right? So BH4 is also called tetrahydrobiopterin. It's not, you don't need to know the names, but like if you were to recognize them, let's recognize them and secure that tetra is for four and dihydropterin is two. So tetrahydropterin is a cofactor for alanine hydroxylase. And why is this important? Because if we need a higher amount of tyrosine, our body requires higher amounts of tyrosine. Something that catabolizes and allows BH2 to reform to BH4 is something called dihydropteridine reductase. Basically, the steps here, it's going to take BH2 and NADH, and when it takes BH2 and NADH, it's going to use the enzyme dihydropteridine reductase to form BH4 and NAD+. Okay, so yeah, so that's about it. Now let's also talk about PKU. Okay, now we're already almost done with the just like you can if you understood the first part, you can understand the rest because this is all built on them. Okay, so phenylketonuria for starters, or PKU, it is basically a deficiency, as we said, of what enzyme? Phenylalanine hydroxylase. Bravo. And this is gonna deficiency of this enzyme is gonna lead to a few things. It's gonna lead to high phenylalanine, and it's gonna lead to low tyrosine. And because of that, it can be a problem mainly with the enzyme itself, okay, or deficient BH4, because we said BH4 helps with phenylalanine hydroxylase, and maybe it can be the problem within the enzyme. Both of them actually differ with like some things that we need to talk about, and we'll talk about it now. So for starters, apart from that, شو اسمه this here phenylalanine can accumulate, which is then diverted instead of forming tyrosine, it's going to form phenylpyruvate, phenylacetate, phenylactate, phenylacetate. Okay, and this is all going to appear in urine instead of our normal شو اسمه levels. And symptoms, okay, obviously will involve a musty smell in natural excretions of body, like smell, sweat, urine. There's a musty smell, okay? Apart from that, you're gonna have also CNS problems like mental retardation. And finally, you're gonna have blue eyes and fair hair. And this is mainly because we have low tyrosine. And as we said, tyrosine kind of contributes to forming melanin. So if we have low tyrosine, we have low melanin, and low melanin basically means you're gonna not have as much expressive features like green eyes, brown hair, and all of that. You're gonna have blue eyes and fair hair. And yeah, this is very important. Please focus on the symptoms. They're so asked on about, especially in SAQ. I guarantee that this might come as an SAQ. If not, it will come as an MCQ. Okay. Apart from that, let's also talk about. Let's just go over it again. The phenylalanine, instead of converting kind of into tyrosine, it's just gonna form phenylpyruvate, phenylactate, phenylacetate. This is important because this has also been asked about. Okay. So let's also talk more about it and its diagnosis, its treatment way. Now, how do we treat a PKU? Logically, if we want to treat PKU, we said we have a lot of phenylalanine. So the first thing we want to do is we want to prevent the patient having more phenylalanine and contributing to his symptoms, right? If we have less phenylalanine, it is going to be the good part if we have a defective enzyme, right? And this is basically telling us the right titration of phenylalanine. We avoid aspartame. Now, for no, there's those. Please don't confuse aspartame with aspartagine or the enzyme we talked about in leukemia. This is actually something we're found, and it's basically a food sweetener. And because it's a food sweetener, it is found to have methyl groups of phenylalanine and tyrosine. and it is important to avoid them. Like if you're gonna eat from outside, because they're not as شو اسمه, they're gonna basically cause these symptoms and increase phenylalanine. So basically, this is under this technically. Apart from that, you have to monitor phenylalanine levels, obviously. And apart from monitoring the levels, we need to شو اسمه, ensure that tyrosine, because tyrosine شو اسمه, tyrosine levels are low, it kind of becomes an essential amino acid. Even though it's not an essential amino acid, it becomes because there's low tyrosine, right? And so we need to give this so the patient doesn't present with like the it's just not low melanin, catecholamines, because these are all important, right? Okay. Now, another thing I want to talk about and move on over, that phenylalanine, or fun fact is that phenylalanine levels are actually measured 2 to 3 days after, like, birth check for PKU. And yeah, that's just to mention if they ask, like, SAQ, like, what is the first thing you must do to a baby or whatnot to check for PKU, to measure phenylalanine levels after 2 to 3 days. Okay. Now, another problem relating PKU is actually going to be deficient BH4, which you already talked about. And this is due to often a defective, and most often a defective dihydropteridine reductase, because this is the second enzyme that might be affected, which might not allow for BH4 formation, right? And for this reason, symptoms are going to include this, like the ones we already talked about, mental retardation, all of that. But here you're going to also notice that there's decreased dopamine and decrease the serotonin synthesis. And because of this, if you guys remember, if we have less dopamine, we're gonna have increased prolactin. Because if you guys remember, we said that prolactin is not stimulated by another hormone; it is actually inhibited by another hormone, which is dopamine. So if we have less dopamine, we have high prolactin. Okay. And also, the treatment here is also the same as these for if we have BH4 or dihydropteridine reductase inactivity. But the thing here that we need to monitor or also give is to give supplemental BH4, obviously, dopamine to prevent increased prolactin, and serotonin, so our body can continue. Okay. Now let's link this with tyrosine. Okay, like let's talk about tyrosine and why tyrosine is linked شو اسمه, to dopamine synthesis and all of this. Okay, so for starters, we have this whole table, and I'll explain it properly, and which all of the enzymes for all of this. Okay, so if you guys remember in ERP as well, we talked about how tyrosine forms L-DOPA. L-DOPA forms dopamine, and dopamine forms epinephrine to epinephrine eventually. So, let's talk about. I'm gonna mention all the enzymes and the cofactors you need to know for each. So tyrosine, to be converted to L-DOPA, it's going to need something called tyrosine hydroxylase. It's gonna remove a hydrogen, and it's gonna form something eventually. Here, if you notice, there's a removed here. It's back there. I didn't show the full picture, but here there's a removed hydrogen. And tyrosine hydroxylase, because of this, the cofactors here that are going to be supporting it is vitamin B4, BH4, and NAD+. Now, it's very nice because vitamin B4, BH4, and NAD+, you don't really need to know it, but it's there to understand it. Because شو اسمه, it's also a byproduct with BH4, right? So this is easy to remember. L-DOPA will form dopamine through dopa decarboxylase. Sorry, this is supposed to be an L. Dopa decarboxylase is just gonna basically imagine شو اسمه, it's gonna remove a carbon, and it removes a carbon molecule. It's gonna eventually form اللي هي the dopamine, okay? And the cofactors here are vitamin B6 and carbidopa. And we're going to talk about what carbidopa is and L-DOPA is in a second. But just remember this in a B4, B6, and dopamine to norepinephrine is just metabolized through dopamine beta hydroxylase. Okay, so dopamine beta hydroxylase is going to remove also the hydrogen. And when it removes the hydrogen, it's going to eventually, form just norepinephrine. Now, the important thing here is the main cofactor here is vitamin C. So B4, B6, then vitamin C, and then finally here, yeah, it's not really important, but I guess if we do have to know it, norepinephrine is converted epinephrine through PNMT, an enzyme called PNMT. It's a very long name, but I don't think we need to know. It's not that high. And its coenzyme here, or the cofactor, sorry, is called SAM. Yes, the SAM that is the major methyl group donor. Okay, and that's about it. Okay, so what are L-DOPA and carbidopa? L-DOPA and carbidopa are very important, and they're going to be very important clinically when we talk about it. Carbidopa, okay, it is mainly a medication, and it is combined with levodopa to treat for carbon. Oh, I'm sorry, I'm supposed to start here. Okay, also L-DOPA, also called levodopa, is basically a precursor for dopamine. Okay, So if we have levodopa, L-dopa, it's basically a precursor for forming dopamine, and carbidopa is a medication used with levodopa, okay, to treat for Parkinson's. Okay. So if we were to give patient carbidopa, we're gonna notice that L-dopa is gonna be converted to dopamine, and yeah, which is why we're gonna talk about it now. And which disease? Parkinson's disease. Parkinson's disease, for starters, okay. Apart from the major symptoms like neurological disorders and all that, obviously, patients are going to experience loss of dopamine producing cells. So we don't have that much dopamine in Parkinson's, okay. We took this also in ARP, but because of this, generally اصلا dopamine cannot pass the blood-brain barrier, and because of this we need to have شو اسمه we need to have something important here, which is why we administer L-dopa. L-dopa is the most common treatment, and it helps treat for Parkinson's because if we give L-dopa, it's able to pass through blood-brain barrier and شو اسمه help be the precursor for dopamine, so it can produce dopamine in the brain. So, and another important thing here is that شو اسمه we need to understand that carbidopa inhibits peripheral dopa decarboxylase to enable central to be heightened. So we have outside, like مش outside the brain literally, but like other regions, we have like we have شو اسمه dopa decarboxylase to form شو اسمه eventually our dopamine صح. And there are other regions also, but mainly if we want to give شو اسمه carbidopa as medication, we need to understand that carbidopa will inhibit peripheral dopa decarboxylase, and the reason it does this, so it can allow for main and central activity within the brain itself to increase dopamine synthesis, because at that time we're going to have low dopamine and we need dopamine ASAP to prevent increased prolactin. Okay. And apart from that, here's a drawing I also borrowed. It's a really good drawing, in my opinion. Here you have peripheral and central. Levodopa, شو اسمه, if you have dopamine, it cannot pass through to centrally. So شو اسمه, levodopa is the one that can be past the blood-brain barrier. It's then gonna dopa decarboxylate toward dopamine. Here, dopa decarboxylate can also form dopamine, but it's inhibited by carbidopa to prevent شو اسمه, to increase the activity here more than here. Okay. So yeah. Now we already talked as adenylyl methionine, and as adenylyl methionine, we already talked about it, and it is basically SAM, right? It is basically the major methyl group donor we already talked about, and it's made from two things: ATP and methionine. Okay, so important, very very important. If you guys remember in the شو اسمه, cysteine and methionine catabolism, we talked about how methionine joins with ATP to form SAM, صح? And both are very important because they help generate three things. They help generate adenosine, they help generate homocysteine, and eventually they also help conversion of norepinephrine to epinephrine. Okay, so adenosine is formed. If you guys remember with homocysteine, both of these are formed through SAH, right? And conversion of norepinephrine to epinephrine. Okay, and obviously, شو اسمه, the more we generate SAM, the more we're depleting methionine. So to oppose depletion, we are generating more through B12 and folate. And if you guys remember, because we have the remethylation, right? And remethylation is through B12 and folate going back to generate more methionine through remethylation to generate eventually more methionine for شو اسمه, generating SAM, because SAM is very important to keep converting norepinephrine to epinephrine. So it's like a continuous cycle that's going on and on. So here is what I wanted to show you. Norepinephrine is converted to epinephrine through this very long name, which is why I didn't want to give you it, guys. So it is called also short PNMT, but I put it so you guys can, if you were to recognize it in the exam. And it will give adenosine, homocysteine with B12 and folate is going to form methionine, and methionine is going to eventually form SAM with ATP. SAM is going to eventually help convert as a cofactor for norepinephrine to epinephrine. And that's about it. Now let's talk about other tyrosine byproducts. Okay, other tyrosine byproducts. We talked about how apart there's norepinephrine, epinephrine. There was also thyroid hormone. So the thyroid hormone, the thyroid hormone is converted with شو اسمه, undergoing tyrosine plus iodine to form thyroxine T form. And yeah, that's very easy. And just know that. Because if you guys remember in the thyroid hormone lecture, it was just tyrosine plus iodine, right? Anyways, apart from that, we also have another important thing, which is our melanin. So this is kind of aside from the doctor, because I guess this is so easy. right? So it's basically pigment in skin and the hair and eyes. It's basically how we have expressive features like our beautiful brown eyes and green eyes. We our hair and colorful hair that we have instead of having it be white. And it is basically synthesized by melanocytes and repeating polymer units made from tyrosine. So basically, tyrosine, you have something called tyrosinase. It's very important, by the way. Please remember this because it was asked about before multiple times in TBL. Tyrosine is going to underform dopaquinone, sorry. And then afterwards, it's going to have, شو اسمه, dopaquinone is going to form this repeating unit to form eventually melanin. Okay, but just know tyrosinase is important. Tyrosinase contributes dopaquinone and eventually melanin. So, let's talk about a problem within the tyrosine byproduct, which is mainly specifically in our melanin. So here we have oculocutaneous albinism, also short for albinism, basically occurs due to an insufficiency of melanin. So the reason, if you guys have heard of albinism, we have white hair and all of this is because we don't have that much melanin to help, express the features of our body. And because of this, this might be caused from two things, okay? The most important one, obviously, is tyrosinase because it's what contributes to the formation, which is why I told you guys to remember this because tyrosinase is technically, forming melanin, right? And then tyrosine transporters, also called OCA type 2, which is here OCA type 1, OCA type 2. And basically, here you might have low tyrosine transporters. That's it. So here you have decreased absent melanin, pale skin, blonde hair, blue eyes, increased risk of sunburns, and skin. And yeah, that's about it. Here's a patient, and as you guys can see, all the features here that we're talking about. Yeah. So let's talk about phenylketonuria versus albinism. Because if you guys remember, we talked about how PKU, actually you have fair hair and blue eyes. However, albinism, you also have blue eyes and white hair. So how do we differentiate them? PKU is basically a defect in phenylalanine hydroxylase. Okay, while albinism is a defect in tyrosinase or tyrosine transporters, if you guys remember, so but it's mainly in these two that we're going to talk about. Here, PK has decreased conversion of phenylalanine and tyrosine. Here, it cannot convert tyrosine to melanin. Okay, and causes toxic phenylalanine buildup plus neurological symptoms, while here it causes hypopigmentation and visual problems. Okay, here you have decreased melanin because tyrosine production is low. Here, melanin is decreased despite normal tyrosine. So we might have normal tyrosine, but we don't have as much as a function of tyrosine. Okay, so yeah, this is the I took it from GPT. Hope it helps you guys. And yeah. Finally, oh yeah, and with the same symptoms about the melanin, however, PK includes neurodegenerative disorders as well. Okay, so tyrosine breakdown and alkaptonuria. So let's first split it into two things. Okay, tyrosine breakdown and alkaptonuria, which is a problem about tyrosine breakdown. Okay, remember that phenylalanine and tyrosine are spoken about together. Why? Because both eventually contribute, both are ketogenic and glucogenic, right? And apart from that, they both deliver the same products during breakdown, which is basically fumarate and acetate. Now let's talk about the things here, okay, that about the breakdown. So tyrosine is gonna actually undergo and form homogenetic acid, also called HGA. Homogenetic acid is then gonna form homogenetic acid oxidase by it's gonna form something called fumarate and acetoacetate or acetate, right? And this is basically through the enzyme homogenetic acid oxidase. Okay, I'm sorry about my mispronunciation. And yeah, this is very important because here, homogenetic acid, if you don't have this, it's going to accumulate and homogenetic acid will become bad. So this is the problem that's seen in alkaptonuria. Alkaptonuria basically has a problem within the homogenetic acid oxidase, and it's going to cause a problem and accumulation of homogenetic acid, and it's gonna basically cause polymerization, dark pigment deposited in connective tissues like joints and cartilage. And here, more importantly, avoid phenyl and tyrosine in diet. Okay, so this is probably the most important part about alkaptonuria. Surprisingly, the questions that are more asked about are not like the deficiency of which enzyme, but rather than its symptoms. And there are so much symptoms, so I made sure to put everything. So first of all, we already know there's polymerization, dark pigment, and these pigment will be connective tissue and joints and cartilage. And the classic findings here are dark urine when left standing. So we're gonna see darker urine than usual. We're gonna see arthritis, which is basically large joints and knees and hips. Okay. And finally, calcification of intervertebral discs. Okay. And very important, and I said this here before, but really important, avoid phenylalanine or tyrosine in that because phenylalanine, sorry, is going to form tyrosine. And tyrosine, will contribute and cause a lot of HGA, and this can be bad because it's going to cause all these symptoms. Okay. So yeah. And yeah, please focus fumarate and acetoacetate. This is probably my favorite part of the lecture, all like the whole MOL TBL. Please focus on these two things and that they're formed in both tyrosine and phenylalanine, and they're formed by our homogenetic acid oxidase. So, catecholamine breakdown. We're approaching the end of the lecture, by the way. I know it's long, but please stay with me. Catecholamine breakdown, like how we talked about tyrosine breakdown, right? Catecholamine breakdown is essentially important because it helps us understand how there are diseases that can be caused because of this. So obviously, after our amino acids generate our monoamines, and our monoamines are like our catecholamines, our dopamine, our thyroid, all of these are catecholamines. They are broken down through specific enzymes, as I just said here. And these specific enzymes, okay, are both monoamine oxidase and catecholomethyltransferase. So for those who like, really were tryhards on ERP, you might actually still remember this because these are two important enzymes for breaking down catecholamine. If you guys remember into something called شو اسمه? Vanilli mandelic acid, right? And basically this is through here. So we're going to talk about the different breakdown and different pathways they take to form it, okay? So for starters, here we have the picture. Norepinephrine, if we take شو اسمه monoamine oxidase from norepinephrine and epinephrine, they're going to form something called dihydroxymandelic acid. And then شو اسمه monoamine oxidase is going to then undergo catecholomethyltransferase, and then it's going to eventually form vanillin-mandelic acid. This is one way to do it. Another way to do it is that شو اسمه catecholomethyltransferase is going to form something called norepinephrine and methanephrine. These are important, by the way. And they're going to eventually undergo monoamine oxidase to form vanillin-mandelic acid. So here it's like the byproduct. Okay. So what's so important about here that I need you guys to focus with me on? The most important part about here is that Bismillah ar-Rahman ar-Rahim. Here, epinephrine and norepinephrine, okay, Bismillah. Here, as you guys can see, they have all these pathways, صح? So monoamine oxidase, the way I remember this is that monoamine oxidase always forms, because it's number one, it forms all, of course, it always forms like one product, as we can see here and here. However, catecholomethyltransferase, uh, here, okay, sure, like it's one and then it forms vanillin acid. Here, it's actually taking two steps. So here you have norepinephrine and then methanephrine and then it forms vanillin acid. Okay, so yeah. The second catecholamine breakdown that we can talk about is dopamine. Dopamine here, okay, for starters, dopamine is also converted into monoamine oxidase and catecholomethyltransferase and forms something called homovanillic acid. Okay, so yeah, it's breakdown eventually homovanillic acid. Now, what's special about this is that, there's a specific disease here called pheochromocytoma. Now, pheochromocytoma and شو اسمه, if you guys remember it, was basically a catecholamine secreting tumor. If we talk about this, huh? A it will cause like increased epinephrine or epinephrine, and they eventually lead to episodic hypertension because we have a fight or flight, headache, palpitations, and if you guys remember the five P's, yeah, this is the same thing here that we're talking about, and yeah, that's about it. And just more here, for us to like do pheochromocytoma, we have to check the vanillin acid for increased catecholamine breakdown, or even better. Why I told you guys methanephrine, epinephrine is important because we can check methanephrine levels. We can check it and see it how much levels there are as early as possible. Okay. Now, for the pharmacology, there's only one concept I'm going to talk about, which is basically severely depressed patients. And these severely depressed patients are given basically monoamine oxidase inhibitors. Okay, that's about it. And the reason we give monoamine oxidase inhibitors is to allow for dopamine to be, high. And, our dopamine is our rewarding hormone, right? And because dopamine is the reward hormone, we're going to feel much more happier. We're going to feel much more active and we won't mind like we're just be really happy people. instead of causing dopamine to be broken down and forming homomagnetic acid. So yeah, so some questions, guys, and I apologize for the pictures. A newborn has milky white skin, white hair, and red appearing eye color. This disorder most often results from a defect in which of the following enzymes? If you said D, tyrosinase, you are correct because we already talked about how tyrosinase it forms the melanin. And melanin, if we have a defect in it, it will, it's bad. And don't get confused here between phenylalanine hydroxylase because most of the time we're talking about melanin, and melanin is mainly found in conversion of tyrosine, and rather than phenylalanine because here it has neurodegenerative disorders. Now, overproduction of each of the following hormones by neoplasm is diagnosed by an increase in urinary excretion of vanillin acid. This is very easy. D, epinephrine, that's correct. The genetic engineering has been used to create a strain of mice that are unable to synthesize dopamine. Which of the following genes would be most likely target for inactivation to produce such dopamine-efficient mice? You said E, tyrosine hydroxylase. You are also correct. And yeah, a male infant is diagnosed with PKU on newborn screening. Which of the following amino acids must be present in this diet to prevent a negative nitrogen balance? If you said eat tyrosine, that is correct because we want to keep giving the patient said tyrosine because we have low tyrosine and we cannot administer phenylalanine because the patient has a lot of phenylalanine. And yeah, see here. Oh, sorry. Okay, I did not notice. So just small. Oh, wait. Yeah, that's sorry about this confusion. Here, let's just solve it and read it. Monoamine oxidase inhibitors, they treat certain types of major depressive and phobic anxiety disorders. The concentration of the following enzymes will most likely increase as a result of this treatment. You're gonna have increased شو اسمه increased epinephrine and because you have monoamine شو اسمه you have increased epinephrine, you have increased شو اسمه epinephrine and norepinephrine activity. And yeah, because you have the inhibitor here. Now, this is finally the last lecture, and probably, in my opinion, the hardest lecture. Okay. You really need to focus here because it is very high. There's everything here is high yield, and I'm trying to make it as concise as possible. So let's begin. So here, basically, metabolism of other amino acids. This is basically amino acids in the body, right? That are found. These amino acids, like the other amino acids that we didn't talk about that are going to be metabolized, like how we talked about 20 شو اسمه phenylalanine and other شو اسمه and tyrosine metabolism. Here it's going to be all the other amino acids we didn't discuss. Let's begin. So for starters, tryptophan and serotonin. If you guys remember, we talked about how tryptophan and serotonin are important. Tryptophan because if there's a lack of it, it might cause disease all the way in the first lecture called Hartnupp's disease, I guess it was called. And serotonin we're going to talk about right now. Let's begin. So tryptophan is a precursor for neurotransmitters and a vitamin. Here I'm supposed to write and sorry. And these are three main things. First of all, serotonin. Tryptophan is actually in serotonin. Apart from that, the vitamin you have is niacin. Okay. And the third thing is melatonin. So, the best way to memorize them is serotonin, niacin, melatonin. Okay. The pathway here, the how is tryptophan going to form serotonin? The pathway here is very simple. Tryptophan is going to form شو اسمه 5-hydroxytryptophan, okay? And this is mainly through an enzyme, which is tryptophan hydroxylase. So it's going to be hydroxylated here. And you're going to also have BH4, right? And there's dihydropyridine reductase, okay? That is also responsible for this, and there's as other catecholamines do. Now, apart from that, it's gonna eventually form serotonin. And yeah, just know the step here that it utilizes tryptophan hydroxylase and BH4 dihydropyridine reductase because it helps form BH4 for it. Okay. Tryptophan can also form, as you said, melatonin. There's nothing here. It's just you need to know that the pineal gland is responsible for our sleep cycle. And yeah, finally, tryptophan can also form niacin. And the way it forms niacin is also by a specific enzyme. And it utilizes not an enzyme, but actually vitamin B6. So vitamin B6 here is just gonna continue with the formation, help with the formation of niacin. طب why is شو اسمه why what happens if we have vitamin B6 deficiency? If you have vitamin B6 deficiency, it will explain why we're gonna have pellagra-like symptoms, as we talked about in the other lectures. And it's gonna have a defective tryptophan absorption, and it can lead to eventually dermatitis, dementia, and diarrhea. If you guys remember, I talked about how if tryptophan is not available, vitamin B6 is not available, and vitamin B6 is not gonna cause these things, which is all under heart nerve disease, if you guys remember. Okay. And niacin is also eventually going to form NADH and NADPH. And yeah, that's all you need to know. Now, also another thing that both serotonin and dopamine are broken down by monoamine oxidase, as both are considered the satisfactory hormones, explaining why monoamine oxidase inhibitors are given to prevent their breakdown. Okay. Now, let's talk about histamine and how it relates to heme synthesis. So histamine, and by the way, this is so easy. Histamine here, we're gonna have something called histidine, by the way. Histidine is gonna contribute to the formation of histamine by something called histidine decarboxylase. Basically, we're removing a carbon and we're forming histamine. And what supports it here, the cofactor is actually vitamin B6. Okay, and yeah, like how we just talked about tryptophan and the formation of niacin. Vitamin B6 is also here. So we're going to also notice it's going to release CO2. Okay, so main enzyme histidine decarboxylase, cofactors paradoxical phosphate in active form. And the role of histamine is this is like just mentioned, and we already know it. It is kind of like it mediates inflammatory responses, obviously. And it's also responsible for some supporting gastric secretions and being a neurotransmitter. Okay. And yeah, now let's talk about how heme is synthesized. Okay, heme heme is synthesized through glycine and succinyl CoA. Okay, so histidine and heme are not correlated, by the way. This is another important thing I'm talking about. They're split apart. Okay, so histamine is from histidine, and heme comes from glycine and succinyl CoA. So this is mainly facilitated by an enzyme called ALA synthase. And the cofactor here is also B6. And by the way, for B6, all of them, it is the active form of B6, which is paradoxical phosphate or paradoxal phosphate. And yeah, and then eventually form ALA from ALA synthase. And then it's going to eventually stack up with and form rings to eventually form our heme molecule. After like there's the addition of iron, as you guys can see, right? And yeah, the main enzyme ALA synthase. The cofactors are paradoxal phosphate, and which is the active form of B6. And let's talk about now glutamate and GABA. Now, glutamate and GABA here they're related to each other. Okay, glutamate is gonna glutamate, which is an excitatory neurotransmitter, is gonna also undergo glutamate decarboxylase, supported by vitamin B6. Eventually gonna form GABA, which is gamma aminobutyric acid, also short for GABA. It is an inhibitory neurotransmitter. When we get to neuro, we're gonna understand more about them. Just know this for now. And yeah, the main enzyme, glutamate decarboxylase, cofactors paradoxical phosphate, and glutamate is excitatory while GABA is inhibitory as a neurotransmitter. Okay, so in summary, uh, histamine, heme, and GABA synthesis all require B6 paradoxical phosphate, and also if you guys go back, tryptophan forming eventually niacin, right? Also requires B6 paradoxal phosphate, right? And if we have a deficiency of this, it can lead to pellagra-like symptoms, which are the 3Ds: dementia, dermatitis, diarrhea. Okay? Now, let's talk about the synthesis of creatine. Now, the synthesis of creatine is a bit too you need to focus, okay, because this is really, really important. And it has been asked a lot about, and it is like a concept that you actually need to understand. Take your time in. So creatine, okay, we need to talk about where it's mainly synthesized also. Creatine is mainly synthesized in the liver plus kidneys, okay? We already know this from GIT, but to understand it more, we need to understand what is creatine synthesized from. It is synthesized from three things, okay? Glycine, arginine, and SAM, okay? Now, if you want to mnemonic for this quickly, gas, best thing. You're gonna see a mnemonic at the end. I'm sorry, but basically GAS, gas, glycine, arginine, and SAP. Okay, because here down its methyl group, arginine is responsible here, and glycine is also responsible. So creatine eventually undergoes reversible phosphorylation by creatine kinase, and it's gonna eventually form something called phosphocreatine. And eventually, phosphocreatine is spontaneously converted to creatinine. Okay, so creatine and creatinine, and it is the byproduct released in urine, which can be measured for glomerular filtration rate, if you guys remember in renal. Okay. Another important thing is that, yeah, here it is. So arginine, glycine, and SAM, gas, okay, they're gonna go and form the creatine molecule. If you guys remember some the urea, how three things form it. Here you have almost the same concept, but different, structures. And creatine kinase, reversible phosphorylation, ATP, ADP, and then it's gonna form creatine phosphate or phosphocreatine, where it's gonna be spontaneously turned into creatine non-enzymatically by muscle and excreted eventually in urine, which helps us during fetus. So while arginine also is a precursor for creatine, okay, arginine is actually a precursor for other things we already talked about. So it is a precursor in the urea cycle for our and it's also a precursor for nitric oxide synthesis, and we're going to talk about this, okay? So urea cycle, it's arginine is eventually going to give urea plus ornithine, if you guys ornithine, sorry, if you guys remember, it's mainly through the enzyme arginase, right? So it can help regenerate by the addition of water. And nitric oxide synthase is basically arginine plus NADPH is eventually going to give citrulline plus nitric oxide plus NAD plus. And if you guys recognize citrulline is also in the urea cycle, but nitric oxide is very important here because it is from the enzyme nitrogen oxide synthase. And the reason nitrogen oxide is very important because it acts as a vasodilator, I think. Yeah. So yeah, and then here's the mnemonic gas is a just a very nice simple mnemonic for you guys. Okay, so let's talk about the branched amino acid clinical scenario. Okay, we already talked about branched amino acid degradation and what it forms, and our branched amino acid which are valine, leucine, isoleucine. But what we want to talk about is the small. Here's the cofactor lovely coenzymes for nerds, or the loud class from New Zealand, but they are essentially amino acids metabolized in muscle cells. So this is all revision, and their metabolism is through two things, okay? Transamination, okay, and oxidative decarboxylation, okay? So it's not oxidative deamination here, it's oxidative decarboxylation, which is basically by keto acid dehydrogenase, okay? So yeah, this is important. Please recognize this. So another important thing is that the deficiency of this enzyme results in maple syrup disease. Okay. Now, what is maple syrup disease to begin with? Maple syrup disease, okay, is basically here it says maple syrup is basically a dysfunctional alpha keto acid dehydrogenase, dehydrogenase for oxidative decarboxylation. And basically, to cause things to be accumulated, these are alpha keto acids and branched chain amino acids. Accumulation of this is going to cause increased isoleucine, leucine, and valine. And there are things that we're going to detect. We're going to see a sweet smell in urine, which is why we call it maple syrup. And this maple syrup smell is actually by increased isoleucine. Neurotoxicity we're going to see is actually in leucine. So Valium doesn't really have a role to play here. It has like a role, obviously, it's increased, but the things we want to notice here, increased smell is isoleucine, neurotoxicity, leucine, and symptoms include lethargy, irritability, apnea, and seizures. Now, treatment, finally, very simple. It's basically restricting branched chain amino acid diet, monitoring plasma amino acid concentration, and giving thiamine supplementation. Okay. Now, let's talk about homocysteine and revise it over, okay? So we talked about it again, okay? And I'm doing this because this is very important. So we have methionine, SAM, this is the ATP. SAM is then eventually going to form SAH, S-adenosyl homocysteine. This is by donating methyl group to methyl transferase. And also this is something here which is our, Bismillah, it was our methyl or methionine transferase here that donates it for SAM, right? And apart from that, we have it's gonna form homocysteine by hydrolyzing it. Homocysteine and adenosine حتى, but homocysteine is our main focus here. Homocysteine is gonna be remethylated into methionine by B12 folate and THF, which is tetrahydrofolate, and cysteine, which is gonna be transsulfated through B6 to eventually form pyruvate sulfite. Then it's gonna for sulfite is gonna be oxidized to form sulfate. Okay. The reason I'm repeating this is because we're gonna now talk about homocysteine. So as I told you guys, homocysteine as an application, homocysteine is toxic, which is why we remethylate it and transsulfate it. So, and for this reason, mild elevations of homocysteine can be caused by two things: B12 and folate deficiency, and apart from that, B6 deficiency. Okay. And these are basically associated with increased cardiovascular diseases. And غير هيك, yeah, basically شو اسمه, you're going to notice these, okay? Because B12 and folate are going to cause the شو اسمه, uh, prevent the remethylation here. B6, if you have deficiency, you're going to prevent transsulfuration. Okay, so yeah, another important thing is now we're going to talk about it, which is homocystinuria. Homocystinuria, for starters, is basically a defect in homocysteine metabolism enzymes, and this is basically an autosomal recessive. Okay, and the signs are lens desiccation. So please remember this because this is going to come SAQ or MCQ, 100%. And long limbs, chest deformities, mental retardation, blood clots, and early atherosclerosis by myocardial infarction because we have increased cardiovascular diseases. Dietary treatment involves avoiding methionine because obviously if we add more methionine, we're adding technically more homocysteine, which causes, increased homocysteine, and that's toxic. And increased cysteine, now it is essential because we need cysteine to form, sulfite and eventually sulfate. And we have to give vitamin D6 supplementation and the reason not B12 is because B12 is going to give more methionine and then, we want to avoid methionine to begin with so we don't get more homocysteine. Now, here we said you have your thing here that we're going to talk about really quickly, which is basically methionine from SAM, homocysteine. Homocysteine is basically by methionine synthase, where it's going to form, folate. It's going to come N5-methyl tetrafolate. It's going to form THF through B12. And here's just through methionine synthase and homocysteinase is going to form cystathionin and cysteine. Here they have cystathionin synthase. This is not really important, but to be honest, if we're talking about the classic homocystinuria, like the most important part here, it's basically a deficiency in this enzyme over here. So it's going to cause here the long arms, chest deformities, and yeah. So basically classic homocystinuria is mainly caused and found as a problem in cystathionin synthase, B6. So let's talk about propionyl-CoA pathway in disorders. We're almost done, guys. Inshallah, هلا we finish. Let's talk about it. So valine, isoleucine, methionine, threonine, and alt-chain fatty acids all eventually give you propionyl-CoA. We already talked about this. Propionyl-CoA can then be converted to succinyl-CoA, eventually to major reactions, which is either going to become propionyl-CoA, which is going to become, methylmalonyl-CoA through propionyl-CoA carboxylase, biotin, as we said, B7. Methylmalonyl-CoA is going to form succinyl-CoA, and then this is basically from adenosylcobalamin, which is basically an active form of B12. And methylmalonyl mutase here, which is supported by this, obviously. And yeah. So let's talk now about the disorders. Now, deficiency of either enzyme is basically going to cause two things, okay? So here you're going to have general symptoms, okay, which is basically, and you're going to have general treatment. The general symptoms here are neonatal metabolic acidosis, poor feeding, vomiting, lethargy, possible neurological deterioration, while treatment here is specialized low-protein diet, valine, isoleucine, methionine, and threonine, okay? And yeah, these are mainly here because these are the contributing to propionyl-CoA, okay? So yeah, apart from that, finally, let's talk about the deficiency of each enzyme, what it does. If we do have a deficiency in propionyl-CoA carboxylase, we're not propionic acidemia. Propionyl-CoA, we're gonna have like a problem with propionyl-CoA carboxylase. We might have low biotin, and this is gonna cause increase of this. It's gonna cause propionyl-CoA carboxylase, or here. Clinical features: metabolic acidosis, lethargy, vomiting, hyperammonemia, neurological complications, and yeah. Diagnosis: elevated propionic acid in blood or urine, and treatment is protein restriction in carnitine. Okay, this is important. Please remember this. So you're gonna restrict protein, شو اسمه, or you're gonna restrict carnitine. Okay. Methylmalonic acidemia is when you have a problem in methylmalonyl-CoA. So here you have methylmalonyl-CoA mutase, which is basically maybe low B12, and it's gonna basically cause increased methylmalonic acid, and it's gonna cause similar symptoms here, and the cause is just basically deficiency or defective B12, right? And basic diagnosis is basically elevated methylmalonic acid, and treatment is restricting carnitine as well. However, also supplying vitamin B12 to B12 responsive patients. Okay. And importantly, both can cause severe metabolic decompensation. Okay. Because both also cause the same symptoms and clinical features, almost have the same treatment. Okay. Now, let's finally get along with this and finally finish our MOL crash course. A 6-day newborn who is being breastfed is brought to the ER because he is unresponsive. He has been vomiting and is lethargic for the past 2 days. Examination shows an unattended infant with hypertonia and muscle rigidity. A peculiar odor and burnt sugar is noticed in his diaper. The plasma concentration of the following amino acids is most likely responsible or increase in this patient. Answer is C, leucine, because we already talked about how here when you have increased leucine and bismillah, isoleucine, all of this, right? It will cause the شو اسمه, the maple sugar disease. And a maple sugar disease, you're gonna smell like burnt sugar, right? And muscle rigidity and all of that, right? Yeah. So, a 5-day-old female newborn is breastfed and brought to the ER because of a 2-day history of poor feeding and vomiting. Physical examination shows lethargy, dehydration, hypertonicity, and rigidity of the extremities. There's a sweet odor of maple syrup in a newborn diaper. Laboratory studies showed metabolic acidosis, ketosis, hypoglycemia. Following the treatment of this patient's acute condition, long-term therapy is begun. This therapy is most likely reciprocal to the following. Step B, because branched chain amino acids, leucine, isoleucine, valine. Guys, I'm gonna like go over them quickly. You can just read this and pause because there are like a lot of questions I put because there are 2 lectures. Okay, so let's begin. And the answer here is B, methionine. Okay, basically it's telling you here how there's the conversion of homocysteine to cystathione. Basically, it's gonna tell you, the girl has increased serum concentration of which of the following, and it's gonna be methionine because we have increased, homocysteine and methionine conversion eventually. And here the answer is C, methylmalonyl-CoA mutase, because we have increased methylmalonic acid. So methylmalonic-CoA is the problem here. Okay, yeah, here. And the answer here, if you got it, it is actually B and D. Okay, this was a question that got double keyed also. If I repeated this question because it got double keyed in the first lecture, and here it's supposed to also get double keyed because B9 and B12 both contribute as it's folate and B12, and also there's another factor if you guys remember, which is also THF. And the answer here would be tryptophan because tryptophan is responsible for Bismillah, uh, the precursor of serotonin, right? And yeah, that's it. Creatine phosphate is critically important, and it requires the input of the following, and it would be A, arginine. Okay, the reason arginine is because it also has other things like شو اسمه, glycine and SAM, S-adenosylmethionine, and if you guys remember the mnemonic gas. And finally, the last question. And finally, the answer is C, because if individuals have classic PKU, the amino acid that becomes essential is tyrosine because we're not receiving as much tyrosine conversion because phenylalanine hydroxylase is, defective here. And finally, thank you guys so much. I know I have just spoken so much. I promise this is the end. Good luck on your TBL, guys. You've got this. You can take this as your final exam study حتى. But inshallah, Hassan Abdulaziz has a surprise for you guys. And yeah, I wish you guys all the best on your TBL. I wish you guys the best on your MSK lectures. Uh, yeah, Assalamu alaikum.